Every expectant mother knows that what she eats impacts her baby. And now research shows that is also true for our cows. Maternal consumption of ReaShure during late gestation had a positive effect on the in utero calf, setting her up for better health and potentially even higher milk production once she joins the milking string. Learn more at balchemanh.com/launch and launch your herd for life.
So I'm going to talk about birds today, and I'm going to try and build a case for the potential of epigenetic manipulation in poultry as a food animal species. And I'm going to point to it in a bunch of different directions. So I'm going to remind you, because I know most of you don't work in poultry. I know this is primarily a ruminant meeting, but that's okay. Remind you how that industry is structured. Talk a little bit about the genetic perspective, and thanks, Chad, for getting that started. And I'm glad Kevin showed that picture of the agouti mice, because I left that one out, and that one's an important one for folks to see. But just to give an idea of a genetic approach to try and figuring out what's missing and how epigenetics may be playing a role. Talk about imprinting in birds, which is thought to not happen the way it happens in mammals. Some early work in my group in collaboration with Rosalina Angel from the University of Maryland. Some observations in multiple species about long-term impacts of the environment on intergenerational and transgenerational effects. Some other observations in poultry, and I'll give you some other bird examples, not just birds that we eat. And then actually circle back to a little bit about what Kevin talked about one-carbon metabolism, and then where we can go from there. Okay, so just a reminder, much of the improvement in poultry has come, I have to wear my genetics hat right now, has come from genetic selection. And we went from birds that you probably wouldn't choose from a supermarket in the 1940s and 50s to the modern bird, and these are very different animals. They're the same species, but they're very. I mean, it's not that different from a dairy cow from a beef cow, right? Because they have very specialized selection. One thing that I want to point out to you, and I'll come back to it later, is in the meat-type birds that those birds have very low reproductive efficiency compared to the layer birds, who will produce 4 times more eggs than a broiler breeder will. Don't forget that the genetic potential is finite, and that we are helping our animal choose how it's allocating its resources. And so depending on where that animal might be spending its resources, we can influence the production system that we might be interested in. That might be for meat, or it might be for eggs. Okay, so here's the structure of the business. We have pedigree companies producing elite lines at the top of this pyramid, and at that point, one individual hen ultimately can contribute to 4 million meat-type birds on the ground several generations later. And what's happening is, make sure I can get the pointer going there. What we got happening is we have selection here at the pedigree line. Those produce great-grandparent lines. The great-grandparents are then crossed, and usually this is a 4-way cross, so we take advantage of some of that heterosis that's happening, or a hybrid vigor, to produce this generation referred to as broiler breeders. In this slide, it's referred to as parents. The practical management strategy to produce enough eggs to generate this 4 billion chicks here at the bottom of the pyramid is a result of restricted feeding of these animals. If they were allowed to eat ad libitum, they would become too large and not produce enough eggs to supply the needs for the industry. And just to give you an example, these animals can be fed as little as 45% of ad libitum intake. So don't forget that, because I'll come back to it a little bit later. Okay, so I wanted to talk about a genetic clue as to what's happening and how important epigenetics might be. And I just want to give you this example of a study that my group did years ago working with Paul Siegel from Virginia Tech, which was kind of fun. You could go back to your undergrad days. But we took advantage of lines that Paul had been selecting since the 70s for antibody response to sheep red blood cells. So essentially he took, and I think I put it in the next, I'm going to skip over this. So this slide is, he took a population, immunized them with sheep red blood cells, took the highest responders and the lowest responders, and continued to select in two different directions, with the 7 highest males and the 28 highest females, and the 7 lowest males and 28 lowest females. And he did this for, and I think we're on generation 47 now. I'm going to go see him next week. But he ended up creating these 2 lines, the high antibody selected line and a low antibody selected line. Let's see if I can go backwards. So here's how those generations separated. And what you can see is at a particular stage of development, I think it was 32 days is the time point when you immunize them with sheep red blood cells, you get about a 14-fold difference in immune response. So what we did was we made an F2 cross of these 2 lines and tried to figure out what the genetic basis of that response to sheep red blood cells was coming from. And this is a whole genome association study in which we're trying to identify parts of the genome where we could see signal where it was associated with antibody response. And so the little peaks are not that important. What you can do is you can sum all those peaks, and what we saw was the MHC, which we know is a huge player in acquired immunity, was only responsible for about 10% of the variation in immune response that we saw. And then all those other spots across the genome represented about 30% of the variation we saw. So where is this other 60% of the variation coming from? It's not in the markers that we were testing. It's not in MHC. It's coming from somewhere else. And so is that epigenetic? Maybe. That's where we're going to point our eye. All right. And I think I enjoyed Chad's description of this. This is what I show my students when I teach animal breeding. When I taught animal breeding at NC State, I'm not going to do that anymore at West Virginia. But the phenotype that we care about is this mathematical relationship between genotype, the environment, and the epigenetype. And what we've learned is that these other components, the environment and the epigenetype, play larger roles. And I always like to write environment really big because that's a huge factor in how the phenotype gets translated. Imprinting, I mentioned that I wanted to talk about that briefly, and I'm just going to give you one slide, and this is a relatively old study. We're looking at the effect of imprinting in a very famous mammalian imprinted gene, IGF-2. They went as far back in placental mammal evolution as they could with this South American opossum, found that imprinting was happening in a parent of origin effect in primitive marsupials, but not happening in birds. So that level of epigenetics probably isn't happening in poultry species, so we don't have to really worry about it. I wanted to give you an idea of where my lab group's origins were in the idea of epigenetics, and that is in this intergenerational observation that we had, that by feeding restricted amounts of a particular nutrient, in particular calcium and phosphorus, we saw improvements later in life. We tried to understand where that was coming from. So this first study, working with Rosalina Angel at the University of Maryland, we treated birds with diets low in calcium and phosphorus. And so if we have a control diet here at the top, we have a challenge diet here in the middle. So this was fed a control diet, normal calcium and phosphorus for the first 19 days, and then fed a deficient diet. And then we had another treatment where we had a deficient diet early and then hit with the challenge diet late. And what we looked at was, what's the difference between these two treatments? The control restricted and then the conditioned restricted. And what we saw was that there was an improvement in the utilization or the absorption of calcium and phosphorus from the diet when we did analysis from the contents. So that sort of, the light bulb went on. We thought, well, how can we manipulate this, shorten the exposure of deficiency, take advantage of it in a more commercially relevant scenario? And so we were able to manipulate our conditioning diet down to 90 hours. So the first 4 hours of a life post-hatch. And so we were feeding birds diets very low in calcium and phosphorus for the first 90 hours. And then after 22 days, essentially eliminating any additional calcium and phosphorus from the diet. And then what we saw was out here at the end, when we compared this diet at the bottom, which had the conditioning early in life, to this diet who was only hit with the challenge diet at the end, is we saw improved efficiency, improved bone mineralization, calcium absorption, and phosphorus absorption. So where is this coming from? So we had to dig in a little deeper. And what we saw was that there was alteration in the expression of the phosphorus transporter in the small intestine of these animals that we were conditioning. So the birds that were conditioned, which you would see here in this low diet, had about 2 to 3-fold increase in expression of this phosphorus transporter. We weren't sure what the mechanism was when we were beginning this work, and so we did a lot of literature review, and we also stumbled across what Kevin mentioned about the Dutch hunger winter and the fetal origin of adult disease hypothesis, and I'm going to give you 2 examples of that, of what the mechanism might be. And we started looking at DNA methylation. I'm going to skip over that because we talked about it a little bit. The other sort of very famous example of intergenerational effects on methylation and phenotype is the example of the honeybee, where the queen is fed royal jelly, which alters the expression of the DNA methyltransferase 3. And so the queen has a very different DNA methylation pattern than the typical worker bee. And so we use that as the basis of our hypothesis to investigate and discover what's happening with differences in DNA methylation in our dietary conditioning studies. I'm going to talk a little bit about, I mentioned this earlier, about long-term effects of the environment, and I put diet in parentheses because that's the one that we most of us in this room care about. And I like this slide that Chad sort of referenced this scenario. And this is from Harvard Magazine, and I can't find a reference for it. I mean, I've looked, but I can't pull it out. But I love it because it shows very clearly in a very colorful way that an environmental exposure in one generation, in this case, this mother is smoking, is also not only impacting her body, but her developing infant, as well as, if it's a girl, it's developing eggs at the same time. And so the observation has been that there is a long-term multi-generational effect of that environmental exposure, which causes long-term health impacts from both grandmother to mother to granddaughter. Similar sort of idea is potentially possible in birds. Because birds don't have a placenta, they do have an external manipulatable, you can think about it, the mammalian equivalent would be an external uterus, right? Because you have this little package that we can manipulate and do things to that you probably can't do very easily in mammals. But if you do an environmental exposure to a hen, you do expose her, you expose the developing embryo, because she's depositing resources in this egg as it's produced, but also in that developing embryo are the gonads for the next generation. The only alternative in this case is the sire is only going to have effects transmitted to one generation down. And I will say some of this work that I'm going to talk about is from a group at INRA in France, and that's a particular reference. I wanted to mention a few other factors in the environment, and most of us are familiar with these environmental toxins across the top of this panel, but the ones that we care about are probably at the bottom. nutrition, and specifically high fat and caloric restriction, temperature and drought for plants, and stress and trauma for animals. Have long-term, there's evidence, both epidemiological and some the beginnings of epigenetic evidence, either through DNA methylation or other epigenetic mark measurements that across multiple species, including the ones that we care about in food production. So just wanted to give a couple of examples of other effects in poultry, and I'm going to show you the first one that I discovered in the literature that goes back to 2007. And this is an example of a trait that was passed from one generation to the next. And this is a group out of Europe, and they were trying to understand the effect of stress on learning. And so the model was the chicken, and the stress that the birds were exposed to was random light pattern. So a random lighting in the room. And if you know our ag animals, they like nice, consistent photo periods. So when you jumble that up, it is very stressful for them. So what happens is you ask the bird to find the food along a long corridor. And so they measure how long it takes for the bird to figure out that it has to walk down the hall and turn to find its food source. And what they discovered was that the stress caused a delay in learning. What was interesting is when they went to do their study and reproduce their birds, their incubator had a problem, and they lost all of their control birds in the incubation process. So the lab tech found that they had some of the extra eggs from the stressed birds. Well, let's just put those in and we'll use those for our next generation. And much to their surprise, they looked at all of those birds and all of them had repressed learning. And so they were like, wait a minute, what's going on? And they figured out that even the birds who were not stressed but were offspring of the birds who were stressed had reduced time to actually delayed time in learning. Here's an example of a bird that I haven't personally tried, but maybe one of you in the room has. Zebra finch has been shown with corticosterone exposure early in life to have generational effects through 3 generations. And the example here is that the grandparent was exposed to high corticosterone, and if you look here at the bottom, what you can see is the offspring, so these are the great-grandchildren or the great-grand offspring, had reduced hatch weight, but they also had increased growth rate when their grandmothers were treated with corticosterone or experienced some stress event. Here's one that I really think fits this audience a little bit, and we heard from Kevin about methyl donors in the diet. This is a study that was done by that group at INRA, and they treated one group of hens, these are Muscovy duck hens, with methionine deficient diet. And then they followed the progress of those animals through several generations. And what you can see is the structure of the experiment here is you've got these females that were treated with a low met diet, and then these females that were controls, and they produced the combination matings. And then you end up with 4 groups that had grandmother who either had low methyl in their diet or control, and you have it by gender, so you can see the effect between males and females. And what you can see in this one graph that I pulled out is that the birds, at least for males, the birds whose grandmothers were on a low methyl diet had lower body weight in early life, but the females that had low methyl grandmothers had higher body weight. So there's some gender effects at play here as well. So let's talk a little bit about 1-carbon metabolism and some work that's been done in my group at NC State. And this is primarily the PhD work of Chelsea Phillips with some support from Rosalina Angel at University of Maryland. And our goal was to try and understand if supplementation with methyl donors could have an influence on any traits that we could monitor in poultry across multiple generations. And we were going to use our molecular tools along the way, doing RNA sequencing to look at gene expression, whole genome bisulfite sequencing to look at DNA methylation pattern differences. But we really needed to know if there was an effect on phenotype before we got too far down the spending a lot of money on molecular tests. So what we set up was a trial where the grandparent generation was fed a diet enriched in methyl donors, and I'll give you the recipe for that here in a second. But really it was 10-fold addition of choline, betaine, B12, folic acid, B6, and a little bit of zinc because it's an important cofactor. And then we followed the offspring of those grandparents for 2 generations and measured as many phenotypes as we could. And what we saw was, I'm going to give you the recipe. So here's that methyl donor methylation cycle that Kevin showed us earlier, and they highlighted in yellow are all the components that we threw in because we really wanted to push the system in a particular direction. The formula is here, and so we ended up with about 10-fold of what's typically formulated in a diet for a quail. So it's a little bit higher than for a chicken. The interesting phenotypes that we measured that were different across the generations focused a lot primarily around egg weight. And the one that really stood out that went across multiple generations was the size of the eggs that were produced. There was no difference in the number of eggs, but the eggs' size varied considerably by treatment. Just to give you an idea of generation 1, and this is the number of eggs in each one of those treatments of their parents, what you see is that the birds whose parents were fed the high methyl donors had higher egg weights, and as you can see there's a lot of scatter, but there's a lot of number here of individual measurements, so that helped us with the statistics. And what we saw, if they were fed the low or controlled diet early or for the entire time, their eggs were significantly smaller. When we got out to generation 2, what we saw was not exactly the same pattern. We actually saw that the high methyl donor birds had lower egg weights, and the low methyl donor grandparents had higher egg weights. We're not sure what that means yet, but while we were measuring things, I asked Chelsea to go and look at immune response in these birds, and what she found was those birds who were fed the higher starter diet with high methyl donors had an increased response to sheep red blood cells at 32 days of age, which was interesting because when we started looking at DNA methylation across the genome in these birds of the parent generation, what we saw was about 1% of the methylation sites that we were able to measure were either had increased methylation or decreased methylation. Chelsea went in and looked at, do any of these sites have anything in common? And what she was able to pull out was some pathways, particularly, and we may have lost that on the front screen. I can still see it down here, but you guys can't see it. It's antigen presentation pathway. So let me go to the next slide. Maybe it's a big image. Maybe that's the problem. No, yeah, that's what the problem was. So maybe the folks online saw that, but the people in the room didn't get to see it. So it's essentially the pathway for antigen presentation in the antibody response pathway. So thinking about where we can go, and I'm pretty close to time, I want to talk about what we know so far. And we've seen effects of diet on at least 2 generations, but we need to do additional studies. Why did we work in quail? Because we wanted to do a species that had a short generation interval so we could get to an answer faster than if we did it in a chicken or a turkey or an elephant, right? We wanted to do it in something that had a short generation interval. And we don't know what would happen if we only treated the hens or only treated the roosters. So the effect may be equal if we only fed the hens the additional methyl donors in the diet. What I really love to look at is doing this, having these available methyl donors in the diet with a challenge or a stress that pushes the animal to adapt. And we haven't done that yet. And I think that might be a key to trying to unlock what the potential of this application could be in a commercial setting. But what I want to pose to you is that what's happening in commercial broiler production right now by the feed restriction of the broiler breeders, there has to be some impact in that setting, because that is a stressful situation. If I asked each of you to go at 45% of what you would normally eat every day for an extended period of time, I suspect you might be grouchy. And has anyone, and maybe you have or haven't, has anyone been in a barn of broiler breeders about when the feeder is about to turn on? Because if you haven't, those birds know when that feeder is, and they are very agitated. They're ready to eat, and the feed lasts about 2 or 3 minutes, and it's gone because they're ready to eat. But yet when we go to the next generation, these broilers that are the ones that we actually consume, they're provided food ad libitum. So what situation are we setting up ourselves up for, those animals up for? I think that's a really, has a lot of potential for trying to understand what effects of epigenetic effects might be happening in that restricted feeding management scheme. So one approach that we've tried to move towards is trying to understand and track individual birds by what's happening at the broiler breeder level and what's happening at the broiler level. And we've done that with a collaboration with Martin Zwiedoff at the University of Alberta. And this gizmo, this feeding station, Martin refers to it as precision feeding. And there are systems for cattle very similar to this, in which the bird wears an RFID tag. The birds are trained early to walk up the little ladder into the feeder. They line up and they go in, and the system reads which animal is there. It then weighs that animal, figures out how much that animal should be fed at that meal, opens up an internal door, the bird walks through, is fed, gets to finish its meal, and then the floor tilts and the bird slides out the side, and then the next bird comes in. And some birds like to eat big meals, so they'll go to the feeder 4 or 5 times a day, but then there are other birds who will go to the feeder hundreds, I think as Martin said, hundreds of times a day, but they're not getting fed because they've already eaten what they're supposed to eat. And so to me, that's an interesting observation, is to try and understand the individuality between these animals. One of my last PhD student at NC State has been working with Martin on this project. And what we've been able to do is look at gene expression and DNA methylation in the breeders, how they behave, what their physiological markers are, because Martin has been working on metabolomics. And then we have the progeny of individual birds at the broiler level that we're able to connect. And so we have a reasonable data set, so that's still developing. What I think is really interesting, and maybe this is why I suggested Kevin might think about animals with feathers, is he talked about this, but he didn't use this particular term, is this first 1,000 days of life is thought of in human medicine as to be this period of when things are established, how the interaction of the epigenome and the microbiome and the host all develop their relationships. Well, if you think about 1,000 days within the lifespan of a human, that's about the same amount of time as the first 63 days of life of a chicken, a broiler chicken, that to be specific. And if you know anything about how fast they go to market, the first 21 days is inside the egg, and then the next 21 days or 42 days are outside the egg, and then it goes to market. So we might have the opportunity throughout the entire life of the broiler to do something to establish a novel relationship or a novel homeostasis. I would be remiss to mention the yolk sac in the egg as something placenta-like, because it does things in the egg. It does more things than we ever thought. And this is just one example. This was a review paper published, I think it was earlier this year, by my friends Eric Wong and Zahava Uni. But they had studied all the biological functions observed in the yolk sac, and it behaves like multiple tissues in a mature animal. It does erythropoiesis. It has hormone response. It does nutrient absorption, which is kind of what we always thought it did, but it also does other metabolic processes, almost like a liver would. And so some of those functions are mimicked in the placenta. So there are connections. Here's where I think we have opportunities to intervene, and we're right at time. This is the structure that I showed you in the pyramid, but I put in places where I think we have opportunities. As Clay said, I made the transition up to West Virginia as a division director or department head. And so I changed the word in my vocabulary, challenges or problems, now gets totally replaced by opportunities. That's my new word that I have to make myself use. Because motivating faculty can be challenging, and when you couch it as an opportunity, they seem to move. But all these yellow areas, I think, are potential opportunities that we haven't exploited all of the potential that's there. If we just think about the movement from the broiler breeder generation, where we know we have feed restriction happening and potential stress, to the broiler, we may have opportunities to manipulate incubation. We may have opportunities to manipulate the microbiota. We can alleviate stress. We can induce stress in a different way. It could be that what we do as a management practice by feed restricting those birds primes them and primes their offspring to be hyperphagic, to be eating machines, which is what we want to happen in the broiler generation. If that's true, it would be really interesting to understand what the mechanism is of how that works, because then we could tackle that in other species and perhaps use it to our advantage.
At Balchem Animal Nutrition and Health, we strive every day to deliver results you can see in your animal's productivity and your bottom line. From a smooth transition into the milking string for your fresh cows, to a happy welcome home from your furry friend. From a strong start in your poultry flock, to consistent weight gains for your finishing hogs. We expect to earn your business and your trust with our people, our products, and our science. Our people have an intense passion for your animals and your success. You can count on us for honest, candid advice and practical solutions for your toughest challenges. As the global leader in choline production, chelation, and encapsulation technology, we take our obligation to you and to the environment seriously. Our products are backed by the most extensive and thorough research portfolio, while our business is committed to advancing environmental sustainability and animal welfare. In the end, it all comes down to results. Balchem delivers real results you can count on, results that exceed your expectations, and results that bring true value to your bottom line. Leading the charge to meet the nutritional needs of ruminants, monogastrics, and companion animals, Balchem offers a growing portfolio of nutritional products and a dedication to innovation and industry sustainability. Balchem is here to solve today and shape tomorrow.